So again, chapter 10 is how cells divide. That's also the mitosis process, as known as what? Mitosis. Okay. This is what happens for all your cells. Okay. All your cells do this unless it's a gamete cell. Unless it's a sex cell, something that helps you make babies, then it's.
I don't have one.
What? Gametes?
You don't have gametes?
No, on this paper. Hey, Brooke, guess what?
She can't hear you. She's dead.
Birthday bottle.
So, binary fission is not the process of mitosis. Is actually what prokaryotes do. Because if you remember back, the difference between eukaryotes and prokaryotes is that eukaryotes have a nucleus as well as other membrane-bound organelles. And so because of that, because they have other membrane-bound organelles, God bless you. They actually have to go through the steps of mitosis to separate out the nucleus. Okay. Whereas on prokaryotes, they don't have a nucleus, and so they can actually just take the DNA, make 2 copies of the DNA, and then just go, boop, hey, look, there's 2 bacteria now. Okay. So that's what this diagram is going through. The chromosomes themselves are duplicated and attached to the cell membrane, and then so the cells then separate out. And what's called a septum forms in the middle, and that is where the new cell wall actually forms themselves. The book goes really depth into, and actually I used this picture. I grabbed the wrong picture, but it's on there. Really in depth into how there's a whole evolutionary chain between prokaryotic cells and animal cells, and why we think, again, another evolutionary reason or reason for evolution. If you look at the way the cells separate, because on a prokaryotic cell, this septum right here is controlled by a specific protein. That same division part on the animal cell where the furrow appears, where it pinches off, that is also controlled by a protein. And it's about the same protein. The amino acid sequence is nearly the same. So it's very, very similar. So again, it goes to this whole evolutionary mechanism. Let's see if this is going to work today. Well, basically the idea is this. If you're going through binary fission, okay, prokaryotic DNA is circular. Eukaryotic DNA is linear. You know what I mean by linear?
Straight.
It's a line, yeah. It's one single line. There's a start and there's an end on DNA for us. There is a start and an end, but it's circular for them. Okay? And that starting point is known as the origin of replication, right there, the green part, if I can do it. There. All right? That green part is the origin of replication. That is where replication occurs. Replication just means to copy. So essentially it's like this, okay? If you get your fingers to go like this, your pointer fingers is the origin of replication. Okay, now it will replicate both counterclockwise as well as clockwise. Origin of replication. Okay. It will replicate both ways. Okay, it will replicate both this way. Okay, it'll go clockwise. It'll also replicate counterclockwise. Okay, and so what happens is as it replicates clockwise and counterclockwise, it actually starts growing a second ring out. Okay? And eventually that second ring will actually double, and it'll look like an 8. It'll actually rotate around this way. Okay? Now, that will actually break off apart, and now you have 2 circular pieces of DNA. Okay? So it makes a copy, and eventually breaks those 2 pieces off, and it makes 2 circular forms of DNA. Now, cool thing about prokaryotes is that they have this whole circular DNA, but also prokaryotes also have something called a plasmid, which is just a certain chunk of DNA. We'll look at how we use plasmid later on, but plasmid is the reason why we can use pigs to pull insulin from pigs, the protein to make insulin, and then inject that insulin, that protein, into bacteria to actually allow the bacteria to make the insulin, or other growth hormones and other endocrine type production.
I have a question. Okay, the green and the red dot, what is the difference?
So the green is the origin of replication,
Yes,
where it
that's
starts,
where it starts.
and then
Okay.
the red is actually known as the termination site. Termination site, the site of termination. So where does the replication end?
Okay.
So remember, we're starting at our pointer fingers, and we're going to our thumbs. So our thumbs are the site of termination. our pointer fingers is the origin of replication.
Okay, thank you.
So it's going actually both ways. One actually is the correct way. The other one is backwards, but eventually it works out and it all pieces together. Okay. Let's see if this is going to work. There is a good animation actually on your online component of your book, but this one is another form if it works. Didn't work in A1, so we'll see if it works now. There you go. Ready? Set, go.
Bacterial cells generally increase in numbers by the process of binary fission. The cell increases in length while doubling all of its parts, then forms a cross septum and divides to become 2 cells. 2 cells become 4. Those 4 become 8, and so on. This pattern of increase results in exponential growth of the population. The time would...
So y'all see how like that whole idea, if you don't take that one antibiotic, you're like, I'm feeling better, and you still have like 3 days left of antibiotics, how very quickly you can get sick yet again. Okay? And those ones that did exist are probably now more resistant to the antibiotic. So you have to go from taking things like amoxicillin to start taking cephalexin, to start taking like azithromycin, and basically what you're doing is you're increasing the antibiotic strength. You're increasing its ability to break down the cell wall, its ability to shut down the protein production, but you're also increasing the symptoms. Okay, because like amoxicillin doesn't really affect your intestinal tract. It doesn't kill all the bacteria in your intestine, and so you don't end up with a whole lot of intestinal tract symptoms. But then you go towards something like azithromycin, which is a much stronger antibiotic. It will affect your intestinal tract, so you start having other symptoms. You're like, man, I'd rather just have the strep throat than the massive diarrhea. Okay, it's true. So that's that. This right here is the reason why it's very behoove you, or it's worth your time to take all the behoove. It means benefit. Use that in Phil's class. He'll like it. Behoove. It's benefit.
Beehive?
No, behoove. But it's better for you to go and take all those antibiotics. Same thing when you're cleaning. That's the reason why it says like the 99.9%. That 0.1% can eventually grow back, so that's why it's better to clean all the way.
required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
That's why they said E. coli instead of Escherichia.
So the original first two, though, are the daughter cells.
Yes, the one that eventually breaks off is the daughter cell. Yeah, so this one right here is the daughter cell because this was the original cell.
Wait, okay.
So like later on when we do genetics, when we talk about, okay, here's the parent plant and here's the offspring, we call those the daughter plants, even if they're male or female.
So this is the daughter cell, and you see.
So yeah, I see what you're saying. So
Okay.
the second one, oops.
Yeah.
So this one
Yeah.
is the original cell.
Okay.
These, so it's going to break off, and so this one, it's hard to say which one was the original cell,
Yeah.
but let's say that that one was the original cell, this would then be called the daughter cell.
Okay. Thank you.
So that's binary fission. It happens very, very quickly. It's not a whole lot to it. Again, because of this idea that it does not have a cell membrane. Nuclear membrane. It does have a cell membrane. Some vocab. A somatic cell is a what?
Any cell.
Just stop there.
That's just how I remembered it.
It's any cell. Yeah, somatic cell is any cell in your body. From your head to your toes, it is your somatic cell, except for your gametes. And your gametes are the ones that you use to reproduce. Okay? And later on when we look at cancer, if you get skin cancer, which is a what kind of cell? Somatic or gamete? Skin, right? So it's somatic. So skin cells are somatic. So if you get cancer, if you get skin cancer, and you are pregnant, can you pass that skin cancer onto your baby?
No.
No, because it's a somatic cell, But if you end up getting some kind of radiation in your ovaries, or you start getting some kind of radiation in your testes, and that will then, if it affects your gamete, then that gamete will pass on to your offspring. So a gamete is any other cell, so the sperm and the egg cell. Okay. Now, the other difference is also this right here. This is diploid and the haploid. Okay. What does di mean? The prefix di? 2. So you actually have 2n, or double the number of chromosomes. So we have 23 chromosomes. That's our haploid number. So what's our diploid number? 46. So our haploid number is 46. Now, in the book, it does give you actually a list of specific organisms. And you will notice that those organisms do not, the number of cells does not determine the number of chromosomes does not determine the I guess, a hierarchy of the organism. Okay? Yes.
So diploid is 2 times the amount of chromosomes?
Yeah, so the n value is the number of chromosomes that you have. So, like, you have 23 chromosomes, you have 23 sets of chromosomes. Your diploid number is basically the double of that number. Okay? So a haploid is just a single, the number of chromosomes you would have. Okay? So in your body, and the reason why it's haploid and diploid is because you get a chromosome from your parent, from your mom, you get a chromosome from your dad. Okay? And that is why it's diploid, because like on this picture here, this right here could be chromosome number 1, and this right here is also chromosome number 1. Okay? You got this chromosome from your dad, and this is your mommy chromosome. Okay, so both chromosomes do exist. So technically you have this is one chromosome, but they're homologous. They're the same chromosome. So they're both chromosome number 1, right here. So that's basically one of those somatic celled diploid chromosomes.
Okay, and then haploid is just one of those.
Yes, so when we separate these out, and we're saying, okay, this is going to go into one egg cell, and this is going to go into one egg cell. Okay, it's very random. We'll look at this next time for meiosis. But that one egg cell and that one egg cell will only have one of those chromosomes. Okay, whereas if this is a toe cell, then it has both, and that would then be diploid because it's somatic. Okay. The centromere is just the part right here where it pinches together. And the centromere is also where you find the kinetic core. The kinetic core is like the little hook you would, like the belt loop. You remember I talked about like when you grab someone by the belt and you rip them back real quick, they fly forward, right, during anaphase? That kinetic core is that belt loop that they would hold on to. Okay? That's where the spindle fibers are attached to. The spindle fibers are attached to. Actually, the centromere is actually just a location on the chromosome. The kinetic core is actually where it attaches to. Okay? And the last one is the sister chromatid. Okay? So these are both chromosome number 1, or chromosome number 2, or whatever chromosome it is. Okay? These are normally in your cells. So when you're in just general growth phase, you only have one set of cells. Until your DNA goes through replication, you only have one set of chromosomes. So one for your mom, one for your dad. Okay, so daddy, mommy. When you go through replication, those chromosomes become 2. They make a replication. They make double of them. Because every single cell is going to have one chromosome from dad, one chromosome from mom. So these 2 are homologous chromosomes, but the 2 pieces that are replicated are sister chromatids. Okay? So it's the easiest idea is that like this is one chromosome. Okay? This is the way your cell normally is. So your cell normally has this chromosome and another chromosome from your dad. Let's say pink is for mommy, right? Okay. So you've got that one chromosome. Say this is chromosome number 2. Your body is going to go through, or your cell itself is going to go through what's called DNA replication. That replication is making a copy of that cell. So what it does is it then basically takes this chromosome, it makes a copy, and just like how you would on a computer, it then pastes that copy together. Okay? So it actually takes that and pastes it back. Okay? So it takes a copy through DNA replication. That copy then pastes those 2 together. Okay? So these together are sister chromatids because it's the same chromosome, but they're sisters because they're like almost like identical sisters because they're exactly the same chromosome. Okay, it's going to do the same thing.
So is the sister chromosome the exact same as the original chromosome, or is
Yes. And the sister chromosome is merely just this idea that it's basically after DNA replication, it's the exact same of the two that you get, either from mom or from dad.
okay? So you have an original one, and then you have another sister, and then you have another sister.
Two sister chromosomes?
You have two sister chromatids.
Okay, I didn't know.
Okay. Yeah. So, but yeah, and so it's again. Let's see. If okay, so you have the two sister chromatids, and again, those are exact copies from both parents. Now, the cell cycle itself, this is the one I was talking about, how 90 to 95% of the time, your cells are in G1, S, or G2. Only about 5 to 10% of the time it's actually there. If the microscopes were working a little better, I would actually make you go through and count how many are in interphase, how many are in metaphase, how many are in telophase, how many are in interphase. And you would actually do a ratio, and that ratio would show you that about 90, 92% of the time it was in mitosis, it was in interphase. Okay? So we've gone through each of these, but I'm going to kind of hit them again. Remember, prophase is what disappears? The nuclear envelope. Okay? And then what happens to the chromosomes themselves? They become...
Chromosomes bind to the spindle fibers by
They start to spread out, they start to bind to the spindle fibers, and they actually start to condense. So if you notice on this picture right here, this is actually like prophase, this is almost to be prometaphase right here. Okay? So here you see that they're very thin, they're chromatin, but when they start thickening up, that's when we call them chromosomes. Okay? These are both prophase, but this is technically prophase. This would then be considered prometaphase. Okay, so then you go to metaphase. It goes along the metaphase plate. This is where you would find, this is when the centrioles would occur. Okay, so these little pink dots, those are the centrioles. So then the spindle fibers are attaching to those pink dots. And this line right here, you all see that? Yeah. Is known as the metaphase plate. It's actually perpendicular to the centrioles themselves. Okay, that is where they're going to line up. So metaphase is really easy to see because, again, you're looking at mostly if your chromosomes are all in one single line. Same thing here. Okay, and again, this would be a plant cell. This is an animal cell. Anaphase, that's where you're getting ripped apart. Okay, so you actually will see the arms themselves lean forward. And then finally in telophase is where the cell wall is starting to form back up, the nuclear membranes are starting to form back up. This is where you start having the furrows, as well as the cell plate is what starts to form, and all the cell plate is where the new cell wall will appear. Okay. Again, I'm going through these quick because we kind of went really slowly on these last time. Any questions? This is also the time when cytokinesis occurs. I want to show you a video. Pretty good video. This is a newt cell. This is a lung of a newt cell. Or sorry, the lung cell of a newt. This takes about an hour. They actually knocked it down. They took several pictures. And they knocked it to, it's like a 1-minute video.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
So again, this is only about 5 to 10% of the time of your cell. So this lung cell, this new lung cell, is not normally doing this, but only about 5 to 10% of the time to make a new cell, it has to do this. I like to watch this. Let's see if y'all can do this. I like to watch this without the sound. And tell me if you can tell me the phases. When you see it, tell me what phase it is. Okay. What's that?
That's meta.
No, it's not meta yet. That's prophase. It's prometa. That right there is then, that's metaphase. Again, you'll see how they're like all the centromeres are all clumped together.
Anaphase.
Good. Last one is then telophase itself. Okay. Alrighty. So the mitotic spindles are there to kind of control. Okay. It's kind of like this idea. If you've got a bunch of sheep, okay, chromosomes are not sheep, but they just go with this idea that you've got a bunch of sheep, and the sheep are in a pen, okay, and the pen gets knocked down. You better have some way to control those sheep because sheep are dumb. They have herd mind. One goes one way, they all go that one way. Okay, same kind of idea with chromosomes. So our leashes, our ropes, are these mitotic spindle. These mitotic spindle are a combination of both microtubules and proteins. Microtubules are merely just a basically a connective structure. They do attach to the kinetic core, and then the ones that are not directly is called non-kinetic core microtubules. And they do help the cell itself actually elongate as well as move the chromosomes around. Here's a couple pictures. This is the picture as far as it connecting to the kinetic core, and then also looking at this idea of the metaphase plate, where they're all going to line up along that plate. Here's a couple electron microscope pictures. This is that furrow that's going to appear. So whenever they separate out, these actually will pinch off in the middle. And this is a zoomed-in picture of that cell plate. This is where that cell wall will appear. Okay, and what it is basically it's cutting a hole. It's causing holes to form right in the middle of the cell, and those holes will eventually be surrounded by cellulose, because that's actually where that cell wall is made of. Yep. So tell me, what is that phase right there? Prophase. Okay. What is that phase right there? Metaphase. What is this one?
Anaphase.
So you can't really see it, right?
Yeah.
So it's got to be an interface. Right? Because can you see the chromosomes at all? No. Can you see the chromosomes on down here on these bottom ones?
So,
No.
interphase or whatever compared to prophase. Prophase, you can see just dark chromosomes kind of all around.
Yes.
Okay.
Yes. So then what's that under there?
Anaphase.
Anaphase. What is, now this one, let's see. Not very good. Like this one right here, you notice that you can start to see the chromosomes, okay, but they're not nearly as thick as that one. So I would say that actually is either telophase or prophase. Okay, because the nuclear envelopes are starting to either disappear or reappear. They're hard to determine between the two. But if you truly want to see a telophase, it's actually like this one. That's going to start pinching off, and then also this one right here is also telophase. See how they're very, very close together, and they're separating out right there? That will be telophase. That's because of cytokinesis. Okay? So again, make sure you know how to do that, because that would be like one of those really easy questions that you should be able to get. Okay. That's the one I was talking about, how like the evolutionary, this FITC protein is actually what determines it. You actually will be able to find that protein all the way up to animal cells as far as for going through it. Okay. Now, all the cell cycle is controlled by cellular controls, primarily things called CDC2s and APCs, and what it is basically it's cycling. Okay? Cycling is an enzyme that actually connects to the CDK. The CDK is also known as what's called cyclin-dependent kinase. Think back to kinase. Kinase was always something that helps with phosphorylating. Now, if the cyclin is present, it allows the cell to continue on. So like right here, if it's activated, it allows it to go into S phase. Right here, if it's activated, it allows it to go into M phase. And what these are doing is basically checking. It's checking for specific spots. So you go into G1. It's going to say, okay, is the cell big enough? Does the cell have enough nutrition to go through all the cell cycle? Does it do all the things that's available to go through the S phase, which is when it replicates? It synthesizes DNA. And then it checks it at G2. It says, okay, has the DNA been replicated properly? Is the DNA exactly the right code it's supposed to start off with? And then it goes into what's the M checkpoint, the final checkpoint, and that is actually looking for making sure the chromosomes are attached to the metaphase plate, make sure everything's looking good before it goes into anaphase. Okay. Now, if it comes here and the cell is not big enough, it will make the cell get bigger and bigger and bigger. If it comes here and they notice that there's not enough nutrition, it will hold it right there until it adds. But here, it will actually destroy the cell. Okay? If the DNA was not replicated properly, there's some kind of mutation. Let's say this mutation is on the chromosome number 4, and it causes the chromosome number 4 is actually integral to development of the spinal cord. Okay, for nerve endings. You don't want that bad cell there, so it actually will destroy that cell. Same thing here. It actually will destroy those cells instead of just waiting around. Now, these checkpoints are determined as far as cancer. So cancer is like that person that just blows to the checkpoint. Okay, they're driving down the road. There's a checkpoint. They see it. They keep on driving. And everyone's like, stop, stop. But they got like the big guns out. They're like, stop, stop, right? And then they keep on hitting it, and they keep on hitting the gas pedal, and they're just driving, and they run right through the checkpoint. That's what cancer cells do. Okay, cancer cells they say, hey, I don't care that I'm not big enough. I want to keep on going. Okay? And it says, okay, yeah, we're big enough now. Oh, we're not really, but it's cool. We're going to go through replication, through the S phase. And, oh, wait, nope, there's a mistake. No big deal. We're going to keep on going. Oh, wait, we didn't actually attach properly to the metaphase plate. So now one of our cells has 2 copies of the chromosome number 1, and the other cell only has no copies of the chromosome number 1. So it will continue on. And eventually what will happen is that this DNA will continue to get worse and worse and worse until we start forming cancer cells. These cancer cells are basically all cancer is just an overgrowth of unregulated cells. So when you get, like, say, skin cancer, you know you have skin cancer because of what's called the ABCs. Y'all heard of this before? Okay. ABC stands for abnormalities, the borders, and the color. So if you notice a mole, and it's a very abnormal shape, if the borders are not clean, if the borders are like kind of jagged and that kind of stuff, or if they have an abnormal color, so most moles are like a kind of brownish black color, if they start turning blues or reds or purples, those could possibly be skin cancer. Okay? And the reason why is because basically the cells themselves are not being regulated anymore. They're basically just growing as they want to. They go right through those checkpoints. Whereas and most are caused because of what's called the P53 protein, which P53 is actually what repairs the DNA. If it can't repair it, then it just destroys the cell. But if you don't have that protein, if it's abnormal, then it just goes through and just continues to regulate. It doesn't stop at the checkpoints. Okay? And all those are being controlled by the cyclones themselves.